Hola! Welcome to Innate Immunity 2, an evolutionary arms race between us and the bacteria. We're still in innate immunity. The previous video for this was the first line of defense in innate immunity. Now this video deals with the second line of defense in innate immunity. So here's our flow chart that we've been working with. So innate defenses are also called non-specific defenses. Within innate defenses, there's the first line of defense and the second line of defense. The first line of defense includes surface barriers, and the second line of defense includes internal defenses. And then the adaptive defenses are also called the specific defenses, and they include the third line of defense. And the third line of defense, the cells that are associated with third line of defense are B cells and T cells. Okay, so we are in the second line of defense in the innate or non-specific immune system. So the second line of defense is composed of cells, mostly phagocytes like macrophages and neutrophils. The second line is also composed of antimicrobial chemicals like peptides, complement proteins, interferons. And the second line of defense also includes these major processes like inflammation and fever. So after the pathogen gets through the first line of defense, then the second line of defense will come to the rescue. The second line of defense, the cells and the chemicals and the processes in the second line operate against a variety of pathogens. So that's why they call this the non-specific immune system. So the second line of defense, let's look at the components of the blood and the plasma. So if you take a blood sample and you spin it down in a centrifuge, the blood will separate into these parts. So plasma, and then this little piece in here, it's called the buffy coat that contains white blood cells and platelets, and then this lower portion is comprised of the red blood cells. So plasma, this yellow part here, it's mostly water. It contains electrolytes like sodium and magnesium and potassium, dissolved gases like dissolved oxygen, dissolved carbon dioxide. Plasma contains nutrients like carbohydrates and proteins. And then within that category of proteins, there's a whole bunch of different proteins that the plasma contains, including these proteins called complement proteins. And when you activate complement proteins, they will punch a hole in the microbe. Fluid leaks out of the microbe and the microbe dies. So let's look at that cartoon for complement proteins. So here's our villain, the microbe, and it's got a lot of things sticking out of its cell wall and plasma membrane, but in this cartoon it's got these purple boxes, and it says that these purple boxes are carbohydrate that contain this sugar mannose. And then you can see in step one, lectin is this molecule. It's in your blood. Lectin, it binds to the carbohydrate portion of the microbe. So lectin from you, just freely floating around, binds to this carbohydrate portion on the microbe. And then, once it's bound, it's called bound lectin. Bound lectin will split these two complement proteins into C2 and C4. So we'll take C1, and lectin, doesn't really show how it does it, but lectin, once it's bound to a microbe, really what it's doing, lectin is alerting the complement system that there is a microbe in your blood. So lectin will split C1 into these two complement proteins, C2 and C4. And then C2 and C4, C2A and C4b will combine. So here's C2a, C4b, so then you have some more biochemical magic. They will combine together, and once they combine, they activate this complement protein called C3. And then C3 will split into C3b and C3a. And C3a, it doesn't show it, it's cut off here, but C3a will lead to inflammatory processes. C3b will lead to opsonization and cytolysis. So C3b, when it's acting as an opsonin and doing the process of opsonization, C3b will coat bacteria and make them more likely to be destroyed by the immune system. And then cytolysis, that's just when the bacterial cell will rupture because of C3b. So that's the complement protein story. There's also antibodies in your plasma that can activate complement. So everybody knows what an antibody is. It's that thing in your immune system. It's protein in your immune system that can help destroy pathogenic microbes. So antibodies can also activate this complement pathway. So this whole thing is kind of called a complement cascade or a complement pathway. And once it gets activated, then the complement proteins can destroy the microbe. And then, so if antibodies and lectin both activate complement, that will lead to destruction of the microbe. All right, more defense components of the blood, so in plasma. So this shows, here's a plasma membrane, this brown thing, and then out here's the extracellular space, and then in here is the intracellular space, so inside the cell. And it shows there's this blue thing is called transferrin, so it's a protein that sticks out of the cell membrane. And then this is iron. This yellow ball is iron. So transferrin, this transferrin receptor in the plasma membrane of your cells, binds to iron, and then your cell will do this process, receptor-mediated endocytosis of the transferrin receptor and iron. And then you get iron into your cells. And you need iron in your cells to do a whole bunch of stuff. So these molecules called the ferritins include transferrin. So we just looked at the cartoon of transferrin, and then the ferritins also include lactoferritin, and they're basically iron binding proteins in the blood that will bind iron, and then your cells can take up the iron along with the transferrin or the lactoferrin receptor inside their cells. So these ferritins are for transport of iron and for defense. So microbes that are in your blood, they want iron too. Like iron, everybody wants iron. So the microbes are trying to get the iron. Your cells are trying to get the iron. We use transferrin and lactotransferrin receptors to get the iron inside our cells. Bacteria make these molecules called siderophores to trap the iron that we should be getting. So these microbial siderophores, they're iron-binding proteins, and they have a greater affinity for iron than our own transferrin receptors. So what that means is if you see if a siderophore and transferrin both see an iron molecule or an iron atom, the microbial siderophore will bind it with greater affinity, and then that's less iron for us. Some pathogens, so you can get, if you are a pathogenic microbe and you want your iron, you can get it the way I just explained. But some pathogens can get that iron by punching a hole in the host red blood cells with this molecule called hemolysin. And then hemoglobin and iron are released, and then the microbes can get all the iron. And this is just this is a cartoon of the chemical structure of a siderophore. And then here's the siderophore in a microbe bound to iron. Okay, more second line defense components of the blood, white blood cells. So in your bone marrow, you have this blood stem cell that can make all your white blood cells and your red blood cells. So there's a stem cell that can make red blood cells, and there's a stem cell that can make all your white blood cells and your platelets. So the white blood cells include basophils, neutrophils, eosinophils, monocytes, and lymphocytes. And then it shows here in this part of the cartoon, basophils do inflammation, neutrophils, eosinophils, and monocytes, they all do phagocytosis. That means they can eat a bacteria. And then the lymphocytes are the B lymphocyte and the T lymphocyte, and they're involved in adaptive immunity that we'll talk about later. The leukocytes, that's just another name for a white blood cell. So all these guys are the basophil, neutrophil, eosinophil, monocyte, and lymphocytes. They're all leukocytes, aka white blood cells. And the white blood cells that are involved in the second line of defense are the basophil, neutrophil, eosinophil, and monocytes. And then, like we said, the lymphocytes are involved in the third line of defense, or in other words, adaptive immunity, and those are the B lymphocytes and T lymphocytes. And then you come over here to the left side of the cartoon. Red blood cells are for gas transportation, so they transport oxygen and carbon dioxide. And then the platelets are for clotting and inflammation. So you don't have to know all the details of this. It's just kind of cool. So here's the hemocytoblast, or the multipotential hematopoietic stem cell. And you can get from that stem cell, you can get a common myeloid progenitor or a common lymphoid progenitor. And then the lymphoid progenitor will lead to your natural born killer cells, the B lymphocytes and the T lymphocytes, and the B lymphocytes turn into plasma cells to make antibodies. The common myeloid progenitor will make thrombocytes, those are platelets, your erythrocytes, mast cells to make basophils, and then the myoblast will make basophils, neutrophils, eosinophils, and monocytes. And then monocytes, once the monocyte leaves the blood and gets into the tissue, it becomes a macrophage. So there's a whole lineage of cells for you. Defensive blood cells include the leukocytes, those are the white blood cells. They are directly involved in defending the body against invaders. Okay, so the leukocytes can be divided up into the granulocytes and agranulocytes. So the granulocytes have granules in them. So for example, let's look at this guy, the basophil, this cartoon of the basophil. You can see that they have those dark blue granules. And the neutrophils here, they have, you can't really see it that well in the cartoon, but their granules are bluish purple. And the eosinophils, these guys, their granules are orangey red. And the granules do important things. So they have a color, they have a shape, they have size, but they also do important things. So for example, the basophil granules are released and they cause inflammation. The neutrophils can phagocytize bacteria, and when their granules are released, they can destroy bacteria. The eosinophils have their granules, and their granules allow for secretion of toxins that kill parasitic worms. And then the agranulocytes are the monocytes and lymphocytes. So you can't, they call them agranulocytes, but they really have granules, you just can't see them very well. So these are the agranulocytes. So in their cytoplasm, you can't really see the granules very well. And same thing with the lymphocytes. If you look in the cytoplasm, you can't see the granules very well. And I told you this earlier, these monocytes, when they leave the blood and get into the tissue, they're called a macrophage. The B and T lymphocytes, we'll talk a whole bunch about in the adaptive immune system. And then the natural killer cells, natural born killer cells, as I like to call them, lymphocytes, they function in innate immunity, and they're not shown in this cartoon. Agranulocytes continued, so we're here with these three guys that have conspicuous granules. and then the agranulocytes are over here. So the monocyte slash macrophages, they can either be wandering throughout your body or they can be fixed. If they're fixed macrophages, then they live in a certain area and they don't wander. So a fixed macrophage would include those dendritic cells of the skin. And in the old days, they used to have cells names be people's names, whoever discovered the cells. But they're trying to get rid of those, the people's names for the cells. And so we'll just call them dendritic cells. Alveolar macrophages in the lungs. If you find a macrophage in the central nervous system, it's microglia. If you find a macrophage in the liver, they call it a Kupffer cell. When you do a lab analysis, you do this thing called a differential, which is a differential white blood cell count. And you can see the proportions of these white blood cells. And then you can, from that, you can deduce different diseases. So you should have a proportion like of these guys, neutrophils, lymphocytes, monocytes, eosinophils, and basophils. You should have, each of those cell types should have a proportionate number. So for example, if you did this differential white blood cell count, you'd find the most cells that you saw in that differential white blood cell count would be neutrophils. And the least number of cells would be the basophils. Okay, how do these cells all talk to each other? So let's check it out. Here's a B cell, here's a T cell, there's a basophil, eosinophil, there's a mast cell, and here's a macrophage. How do they talk to each other? How do they let each other know, you know, there's an invader there, they have to crush the invader. Oh, sorry, little neutrophil, I didn't see you. You do it, the white blood cells talk to each other with these molecules called cytokines. And this is a cartoon of all the different cytokines. And you don't have to know, you don't have to know this cartoon. I just put it up to show you how complicated it is. So, for example, in this cartoon, the macrophage can communicate with this neutrophil with interleukin 1, interleukin 8, and tumor necrosis factor alpha. And then the neutrophil can communicate back to the macrophage with tumor necrosis factor alpha, or TNF alpha. So it gets pretty complicated, but these cells do know how to talk to each other. So in this part of the cartoon, the neutrophil secreted TNF alpha. The macrophage, doesn't show in the cartoon, will have a TNF alpha receptor so that TNF alpha can bind to it, and then the macrophage can get the signal. Okay. You can call these, okay, there's cytokines is really the broad category of molecules that can talk to each other, that allow cells to talk to each other. Interleukins is a little more specific name. So inter, as you know, means between. Leukins, that's leukocytes. So inter, between leukocytes. So this is communicating between leukocytes. So these molecules can be called interleukins, the ILs, interleukin 1 through interleukin, I don't know how many they've discovered, how many interleukins they've discovered, 20? I'm not sure. But the role of interleukins is signaling and communication between leukocytes. So interleukins, communication between leukocytes. And then humans have designed anti-inflammatory drugs. Those drugs inhibit the synthesis of cytokines so that inflammatory cells cannot create an inflammatory process, or at least it's attenuated. So there's the interleukins, there's the interferons, there's growth factors, there's TNF, tumor necrosis factor, and chemokines. So all of these guys are cytokines, 1 through 5. These are all cytokines. And so really, I mean, for our purposes, the cytokines are like the interleukins, and then there's specific interleukins and all these names here. Okay, second line of defense, phagocytosis. So right here, and it shows a cartoon of it. Check it out. This is probably a neutrophil, and it is about to phagocytize this green bacteria. So what happened? Oh, ouchie, the splinter went into your skin. So, you know, this is all your epidermis from here to here. Here's the dermis. Splinter goes in. Bacteria were in the tip of the splinter. The damaged tissue, so these cells release inflammatory chemicals. And mast cells, and then cells from the mast cells that are on the inside of the blood vessels toward the tissue side, and then cells that come out of the blood vessels will move toward these inflammatory chemicals, because where the inflammatory chemicals are, that's where the bacteria are. So let's check it out. That doesn't have, oh yeah, step one. Inflammatory chemicals are released because this tissue got torn up. So the inflammatory chemicals come down here, and they can tell these endothelial cells, which are the cells that line the blood vessels, that they need to get sticky. And then neutrophils that were once freely floating in the blood, they stick or marginate. They stick to the side or marginate to the side of the blood vessel walls that are made of these endothelial cells. Look at all the cells. We got neutrophil cells, we've got these endothelial cells, and then the endothelial cells, they're more permeable. So see, let's look over here. These two endothelial cells are stuck together. This endothelial cell and this endothelial cell have made a gap, so this neutrophil can get through the gap. And the neutrophils get through the gap because they want to find the bacteria, because they want to do phagocytosis and destroy the bacteria. So they stick, the neutrophils stick or marginate, and then they do, when they crawl through the space between the endothelial cells, that process is called diapedesis. So, you know, pedestrian for walking, and dia means through. So pedestrian walking through. So the neutrophil is walking through the permeable blood vessel wall, because these endothelial cells have separated. And then there's a chemotactic gradient of these inflammatory chemicals. So can you see there's like one inflammatory chemical, and now there's two, now there's three, and now there's a whole bunch. So that's like a gradient. So a gradient looks like this. So you'd have a bunch of inflammatory chemicals up here, and then you just have a few down here, and the neutrophils will crawl along that gradient until they get to the battlefield, as I like to call it. That's where the bacteria are. And then the neutrophils recognize the bacteria. So they recognize the PAMPs, or pathogen-associated molecular patterns, on the bacteria with the neutrophil's Toll-like receptor. And then when the recognition occurs between the neutrophil and the bacteria, then the neutrophil can do phagocytosis. So it can chew up the bacteria and destroy it. So phagocytes, and in this cartoon it was a neutrophil, so they marginate, and they do diapedesis in concert with emigrating. So emigrating, leaving the blood vessels. Then they do, the neutrophils, well, the phagocytes in general, but a neutrophil in this cartoon does chemotaxis, so it walks along the chemical gradient to get to the battlefield. And then it does a process called adherence, and really the adherence is just when the neutrophil or the phagocyte recognizes the microbe, and then the neutrophil can do phagocytosis. And we'll look at that next slide. So chemotaxis is movement of a cell toward or away from a chemical stimulus. In this case, in inflammation, the phagocyte is moving toward a chemical stimulus. There are chemotactic cytokines that stimulate leukocyte attraction to the infection. So the cytokines, so we saw that. The cytokines include interleukins, TNFs, etc. And they're chemotactic because they form this chemotactic gradient, and that the leukocytes or white blood cells can walk along that gradient and find the battlefield. Then phagocytes use pseudopodia to move toward the site of infection. So all that means is so pseudopodia is false foot. So what they do is right here in this cartoon. Check it out. They'll push their cytoplasm and plasma membrane forward here, and then the rest of it will just fill in, and then this will get bigger, and then they'll make a new little pseudopod or false foot, and they'll keep moving that way. They use their pseudopodia to ingest or eat the bacteria as well. Chemicals that attract phagocytes, so in this cartoon it just says inflammatory chemicals, but chemicals that could attract phagocytes in general could be microbial components and secretions from microbes, components of damaged tissue. So that's what this cartoon is showing. The chemicals came from damaged tissue. White blood cells could also secrete chemicals that would attract phagocytes. Chemotactic factors can attract phagocytes, and all these molecules can attract phagocytes as well. So here's a cartoon that shows phagocytosis. So in step 1, so the phagocyte moves to the microbe, and the microbe and the pseudopodia of the macrophage adhere to each other. So these pseudopodia on the macrophage have toll-like receptors that recognize the PAMPs on the bacteria, and so the bacteria sticks to the macrophage. And then the macrophage, or the phagocyte in general, ingests the microbes into this thing called a phagosome, and then a lysosome fuses to the phagosome. It becomes a phagolysosome, and then the lysosome has lysozyme enzymes in them that will tear up the bacteria inside the phagolysosome, and then you can also, well, and that involves killing of the microbes with the enzymes and other chemicals that were in the lysosome. And then the pieces that won't hurt you of the microbe are eliminated by exocytosis. So the correct sequence of events in phagocytosis include chemotaxis, so the phagocyte has to get to the bacteria. And then you have attachment or adherence, endocytosis or ingestion, you call it either one. Then you fuse the phagosome and the lysosome, and then you get digestion or killing of the microbe inside the phagolysosome. Then the residual bodies are exocytosed or eliminated. Those are the correct sequence of events in phagocytosis. So we just look at it in a little more detail. Adherence. Phagocytes attach to the microbes by binding of complementary chemicals. I told you it's PAMPs on the bacteria and toll-like receptors on the phagocyte. Some bacteria will make these virulence factors that hinder adherence so that the bacteria cannot be recognized by a phagocyte. So for example, the M protein of Streptococcus pyogenes, or a capsule, would also be a virulence factor that inhibits the macrophage from binding to the bacteria. Bacteria are more readily phagocytized if they're covered with antimicrobial proteins. For example, if the bacteria are covered with complement proteins, which we talked about a minute ago, or if the bacteria is covered by antibodies, that makes them more amenable to be phagocytosed or eaten by the macrophages. So anytime you cover a microbe with these proteins, that is called opsonization. Okay, then ingestion occurs. So that's when you endocytose the bacteria, or the macrophage, or the phagocyte in general endocytoses the bacteria. And then this phagosome or ingestion vesicle, it is fused to the digestion vesicle called the lysosome, and that together is the phagolysosome. And then you kill the microbes. So I told you there's lysozyme in the lysosome. So you have to think about the lysozyme is the enzyme. Lysosome is that structure inside the macrophage or the phagocyte. And lysozyme will cleave the bonds between those sugar subunits in the cell wall. Remember NAG and NAM, the peptidoglycan cell wall. Lysozyme destroys that. And you can, with this process of phagocytosis, you can kill bacteria within 30 minutes. And then once they're dead, you eliminate them or exocytose them. So this is a scanning electron micrograph. So here's our hero, the phagocyte. Could be a macrophage, could be a neutrophil. And then here's a yeast cell being phagocytosed by our phagocyte. Major microbial attachment sites for the phagocytic white blood cells include things like a flagella. So if you're a phagocyte and you see a flagella, that could be an attachment site for you. You know, that's so you could kill the bacteria with the flagella. Spike proteins on viruses is an attachment site for phagocytes. Lipopolysaccharide, or LPS, so that's a component of gram-negative cells. And then peptidoglycan, that's what makes up the cell wall in bacteria. Okay, eosinophils. So here's a photograph, a light micrograph of an eosinophil. It's a leukocyte, it's a white blood cell. It's eosinophils. If you have a high eosinophil count in your blood when you do a white blood cell differential, that means you could have a protozoan or a worm infection. It could also mean you have allergies. So eosinophils primarily will attack parasitic worms. So here we go. Here's our cartoon. Here's the eosinophil. Here's the poor worm in green. The eosinophil is dumping all those orange-red granules that have toxins that will kill the worm. So eosinophils can attach to worms. They secrete that protein toxin onto the surface of the worm to kill it. Eosinophils can do phagocytosis like neutrophils and macrophages, but that's not their major function. Okay, let's talk more about the second line of defense, the non-phagocytic killing this time. So these guys don't use phagocytosis to kill. These are the natural-born killer T cells. So this cartoon, here is one of your cells. So, oh, I got infected by a virus. And over here, some tumor cells that could turn into a cancerous tumor. And here's our hero. The natural-born killer cell can kill virus-infected cells, and they can kill tumor cells. So natural-born killer cells are kind of cool. And this is a natural-born killer cell. It's got those dendrites, you know, those elongations, and then it's trying to kill that giant purple thing. Okay, other ways to kill bacteria is through this apoptotic pathway using the perforins and granzymes. So this could be the surface of one of your cells that's infected, or it could be the surface of a bacterial cell. But what happens in this apoptotic pathway is that, well, specifically for the innate immune system, these natural-born killer cells, or NK cells, they have vesicles in them containing this thing, this blue thing called perforin. That's the thing that pokes holes into the bacteria. And then the cytoplasm of the NK cell also has vesicles containing this guy, granzyme. Then granzyme can go in through the hole that perforin made, get into the bacteria, inside the bacterial cell, and initiate this process of cell death called apoptosis, and then the bacterial cells die. Interferon, that's IFN, if you remember the cytokine molecules, and TNF, tumor necrosis factor, those can be toxins, and they're involved in the apoptotic pathway. Okay, neutrophils. Neutrophils can kill using phagocytosis, but they can also kill non-phagocytically. So neutrophils don't always devour their pathogens. Neutrophils have enzymes that create reactive oxygen species molecules that is converted into bleach, and then those kill the bacteria. So let's see, here in A, so here's a neutrophil engulfing this bacteria, and then the neutrophil makes these reactive oxygen species, and then the reactive oxygen species will kill the bacteria, and then the reactive oxygen species will also go outside the neutrophil and cause tissue damage, which is unintelligent design, but the neutrophil did kill the bacteria here on the inside. And then this is just another cartoon of a neutrophil releasing the chemicals to kill the bacteria. And then there are, well, and this also is a cartoon of these neutrophil extracellular traps or nets. So these are nets. They're extracellular fibers, mostly made of DNA, and they can bind up pathogens, and then the neutrophil can dump the reactive oxygen species or bleach onto the bacteria and kill it. If you look at pus when you get a wound, what you're seeing primarily are neutrophils. Okay, more cells in the second line of defense, the dendritic cells or DC cells, dendritic cells. Dendritic cells with their Toll-like receptors can recognize microbes, allergens, pollutants, chemicals from tissue damage. And then they can alert, these dendritic cells can alert the T lymphocytes. And the T lymphocytes come in different flavors, T helper 1, T helper 2. So they've got T helper 17. And then there's also another type of T cell that's called a cytotoxic T that we'll talk about later. But the dendritic cells live in the skin. They bind to the bacteria or the pathogen of interest, and then they leave the epithelial tissue and travel in lymph and present epitopes that they chopped up from the bacteria in the lymph nodes. So this is just a cartoon of the lymph nodes. So the dendritic cell would get here in a lymph capillary, and then the dendritic cell with its bounty, its bacteria, it would travel to any one of these lymph nodes, and it would show T cells. Hey, look, I found this bacteria, this piece of this. Well, I found a bacteria, I chopped it up, and now I'm showing you a piece of bacteria, T cell. Now you go back and try to destroy all the bacteria that have this piece on them. Dendritic cells are in the second line of defense, but this is tricky. The dendritic cells will activate the T cells, which are part of the third line of defense. So sometimes the cells overlap. The dendritic cells in the second line will take parts of bacteria to the T cells in the third line. Okay, here's my Toll-like receptors that I was talking about. So you see down here TLR1 and TLR2 up to, they've got TLR9, remember that stands for Toll-like receptors. And these different Toll-like receptors will recognize different pieces of bacteria. So for example, a bacterial lipoprotein will be recognized by TLR1 and TLR2. Toll-like receptors are on the phagocytes and on natural-born killer cells, and they recognize PAMPs that are on the microbes. And when Toll-like receptors on phagocytes recognize PAMPs on the bacteria, that triggers that phagocyte to react to the bacteria or viral invaders and destroy them. When Toll-like receptors bind to PAMPs, this initiates defensive responses, including apoptosis, which could mean if I'm an infected cell and I do apoptosis, I destroy myself for the good of the community. Nonspecific chemical defenses also include, we've talked about this, lysozyme, defensins, interferons, and complement, and a bunch of other proteins. What about interferon, or INF? So let's check out the cartoon. So here's a host cell. It's like, oh no, the virus got in. Sorry, virus got inside. Virus multiplied itself. But this host cell that got infected was like, all right, I'm infected, I'm done for, but I'm going to send out these alarm molecules called interferons. And then cells that have not been infected down here will bind interferon with their interferon receptor. As always, you have to have a receptor. Well, not always. Most of the time, you have to have a receptor. And then these cells that were able to bind to interferons will set up a series of defense mechanisms that will allow them to be resistant to the viral attack. So now these cells, they were alerted by interferons from their dying buddy over here. And now when the virus tries to get into these guys, they don't succumb to viral attack. So these interferons don't protect the infected cell. That's this guy up here. He's the host cell or the infected cell. He's done for. But he did alert his buddies that there's a virus around. Yeah, this infected cell will die from an apoptotic death.